Thank you all very much for joining us to our rescheduled May CISMA webinar. So the Florida Invasive Species Council's Partnership Assistance and Communications Committee, or FISCPAC, helps host these webinars and support our CISMA's cooperative invasive species management areas, which are, the boots on the ground doing a lot of the invasive management work all across Florida. So if you aren't already tied into one of your local CISMAs, you can go to floridainvasives.org/CISMAs and find out what the local group is and get involved with those. So for today's presentation, we've got our brief introduction right now, and then we will be hearing about the invasive snake lungworm parasite from Jenna Palmisano. Looking forward to hearing about this new invasive species that's across the state here, and then we'll have some brief FISP and CISMA updates to end the presentation. So I'm going to go ahead and stop sharing and hand it over to Jenna.
Awesome. Thank you, Deb. I'm going to share my screen now.
So one more quick thing as we go through the presentation, if you can keep yourselves muted. If you have any questions, feel free to throw them in the chat and we will answer those at the end of the presentation. All yours, Jenna.
Okay, perfect. Thank you for being patient, and I'm sorry that I had to reschedule. But today I am going to be talking about the invasion of Rescheduled orientalis, which is a growing concern for Florida's herpetofauna. So my name is Jenna Palmisano. I am a third-year PhD candidate in Dr. Anna Savage's lab at the University of Central Florida. And so pictured here is a pygmy rattlesnake, and that's actually the focus of my dissertation. But everything that we'll talk about today is tangential to that because Rescheduled orientalis does threaten pygmy rattlesnakes. And so you can see one of the adults crawling out of a deceased pygmy rattlesnake here. You can see everything good, Deb? Okay, cool. Awesome. So just a quick overview of what I'm going to be talking about today. I'm going to give some background on pentostomes, so an evolutionary perspective, what their life history looks like and their diversity, the disease that's associated with infection, who we have for pentostomes in North America, especially in the Southeast is what we'll focus on, conservation concerns, and then some data from the group known as Snake Lungworm Alliance and Monitoring. And then if we have time, I'll just kind of talk a little bit about my dissertation studying the dusty pygmy rattlesnake. And on the right, we have a large Rescheduled orientalis in a Burmese python lung. So what are pentostomes? They are actually crustaceans and they are endoparasitic and they reside in the upper respiratory tract vertebrates. And those are primarily going to be carnivorous reptiles. So Rescheduled orientalis is actually, there's pictured on the right here, and you can see 4 hooks and then kind of the feeding apparatus on the top there. And these animals are hematophagous, so they are living in the upper respiratory tract and feeding on blood once they reach their reproductive stage. So one really cool fact about pentostomes is that they are the earliest recognized parasites in science. So they first found fossils from the Silurian period, which is really neat. And there's a drawing of one of those fossils here on the right. So they've kind of had evolutionary stasis in their body plan where they kind of have worm-like bodies, so that's why we call them lungworms sometimes. So they're vermiform, and then kind of a thicker head portion with a feeding apparatus and also some paired hooks. So across the world, there are 125 pentostome species recognized. There's probably more. It's quite an understudied group of animals. And they occupy a large gap in our crustacean knowledge, both biologically and in an evolutionary sense, because like I said, they're really understudied and they used to kind of be classified as trematodes and other different parasites, but they're actually crustaceans. And so here's just kind of a phylogenetic tree just showing you that recent data, we actually assembled the mitogenome of Rescheduled orientalis, and it just fixes the pentostomes within the crustacean clade. And on the right, you can see some different female pentostomes. Again, they all kind of have that worm-like shape with different head shapes. And in the top left is a Rescheduled. So Rescheduled, they typically have more of like a flattened triangular shaped head. And we'll take a look at quite a few today. And so just to kind of give you an idea, they have kind of funny drawings of their head shapes, but they're all vermiform and have paired hooks throughout their life cycle. So they have 3 stages regardless of the amount of hosts that they have. And they have an embryonated egg, which you can see on the left, and then a larvae, which you see in the middle, kind of tardigrade-like, and then the adult stage, which you can see an example of on the right. So pentostomes are really diverse. They exist on all continents. And there are a couple that we know of that have direct life cycles. So Yellyrella, which is the smallest pentostome, those infect sugar gliders. And so they have a direct life cycle. And then Regardia, which infects a lot of shorebirds, that also has a direct life cycle. And then the majority of pentostomes, like the one life cycle we see on the right here, have 2 host life cycles. And today, a lot of our focus will be on the invasive pentostome, Rescheduled orientalis, which is a little abnormal compared to other pentostomes, and it has a 3-host life cycle, which we will go over. But just kind of hitting on this point again is that pentostomes are really diverse, and there are a handful that are more generalist in nature. So Linguatula serrata is actually one of the only ones that can infect humans as the definitive host. And it's most common in the Middle East, but it's also invasive in Australia. And then Rescheduled orientalis, who's kind of the topic of our presentation today, also has a really widespread host range. So there are, specialists like these 2, the Yellyrella that infects just sugar gliders, and then other ones that just infect shorebirds. But then there's some like Rescheduled orientalis that are super generalist in nature. And so pentostomes have evolved to be pretty low key in their host, where they have surfactants that they cover their entire cuticles with to kind of remain hidden from the host immune system. So often pentostome infections are asymptomatic, especially in those hosts that they have a coevolutionary history with. But often the migration or death of the adults or the larvae moving to the lungs can cause issues where they are having lesions and hemorrhaging or necrosis and inflammation. And in naive hosts, which kind of is the biggest issue here and what we'll get into today here in our Florida native snakes and lizards, it can cause occlusion of the air passages, pneumonia, so lesions of the lung, anorexia, and septicemia, which we have seen in our native snakes. And pentostomes in naive hosts have been a big problem in captive animals across the globe, which are just some examples here on the right. I think the top one was a monkey that was held in, or some primate held in a zoo in Brazil that got, I think it was porocephalus, and then the hyena down in the middle here had armilifer. And so these animals were being exposed within their enclosures. So a lot of zoos, especially in Florida, have outdoor enclosures, and that can be a way that animals can be exposed to infection either from the wild or expose wild animals to infections. And it's also important to note that all cases of human pentostomiasis are zoonotic. There are also some fitness consequences that have been documented from infection. And so really the only species that has that we've looked at this in is Hemidactylus geckos in Texas, with a different Rescheduled species than our focus today, Rescheduled indica. And basically they found that it decreased egg production, it lowered liver and fat mass outside of the breeding season, and it increased the geckos. So infection increased the geckos' recovery time and decreased their CO2 output after activity. So basically this is showing that the heavier load in any load of pentastomes in these geckos is likely causing some type of mechanical constraint. And so that's kind of our background of pentastomes. So if you have any general questions of their evolution or biology, happy to answer those. But now I'm going to move into the native pentastomes here in North America and then switch to the invasive ones. And so we have quite a few native pentastomes. So some that infect turtles, some that infect gators, and then we have 2 native that primarily infect snakes, the Porocephalus crotali, which we see a lot in cottonmouths and eastern diamondbacks, and then Chirocephalus coarctatus, which we primarily see in Nerodia species or water snakes. And the invasive pentastomes we have in Florida in the southeast are Relicella indica, which parasitize geckos as adults, and then Relicella tegus selfie. We know that that has been documented in Texas, but it hasn't been documented in geckos in Florida just yet. And then we have the focus of the rest of the presentation, which is Relicella orientalis, and that infects snakes as the definitive host, and unfortunately other animals too, which we will get into. So I'm sure a lot of you have heard about the origin story of Relicella orientalis here in Florida, but it is likely a spillover event that happened with the Burmese pythons who have co-evolved with Relicella orientalis. And we found Relicella orientalis in a lot of different invasive hosts, which we'll see later on. So it is possible that it's been introduced multiple times, but we don't really know. The best evidence we have right now is the direct spillover from the Burmese pythons, which Dr. Melissa Miller originally documented. And the reason why it's a problem in our native snakes is that compared to Burmese pythons, the pentastomes, or Relicella orientalis, or RO, I'll just refer to it as RO moving forward, is that the infections get a lot more intense in native snakes. So you can see an example here of an intense infection of a pygmy rattlesnake. They can have, not pygmies, but racers can have over 100 females in the lung. I've seen up to 107. And so very severe infections in native snakes. And then the parasites are also getting larger than they do in the co-evolved, or likely the co-evolved host, the Burmese python. And so these infections have been documented to be causing pneumonia and sepsis, and it's been associated with mortality and morbidity events in at least 6 species. And we're starting to see a lot of evidence, both empirical and anecdotal, that this invasion is associated with population declines in our native snakes. So getting into kind of where is RO? Well, it was first documented in these purple counties here by Melissa Miller in 2017. And then we saw a huge jump in 2019 to Alachua in orange, that was a banded water snake, and then Volusia County, which was the first documentations there, and that was in 3 pygmy rattlesnakes. And then Dr. Miller published more of her data, and we saw these yellow boxes here are showing you infected snakes. We saw kind of filling in the map a little bit more. RO was definitely moving. And so I had a big question, me and my advisor in my undergrad, of, okay, how is RO expanding the invasive range beyond the Burmese pythons if they're the ones kind of the culprit of the spillover event? And so we conducted experimental infection studies and found that that RO likely exhibits a 3-host life cycle. And this is an open access publication. If you are interested, I can send you the PDF. But basically what we found is that cockroaches or other coprophagous invertebrates that feed on the feces of snakes or other definitive hosts, they act as a first intermediate host where they ingest eggs and the larvae start to develop in their fat bodies. And then the second intermediate host is a frog or a lizard, and those animals eat the coprophagous invertebrate, and then the larvae start to develop, and they grow about double in size once they are in this host. And there's a question mark next to this small mammal here because we have very little information on the role that mammals play in the life cycle of RO, but we do know that there was a Kenyan shrew documented with larvae, and Melissa Miller reported that. So we don't really know what role they are playing, but we do know that mammal eaters in Florida are becoming infected. So they do play some role. And then the final host is the snake. And so I'll just walk you through this a little bit more. So the eggs are passed with the feces of an infected host, and then a coprophagous invertebrate like a cockroach is going to feed on those eggs. And you can see examples of those eggs in the top right there. And then they'll develop the initial larvae. So this is kind of what the larvae look like under a microscope from cockroaches. So this is from the experimental infection. And you can see even at this stage, they have the four hooked appendages, and then this feeding apparatus is called the buccal cadre. So kind of tardigrade-like, if you're familiar with that body shape. So then an anole or a frog is going to feed on that cockroach or other invertebrates, and then become infected with larvae that are about double in size. And so this is an Anolis sagrii that was in one of our experiments, and you can see all those white kind of stars. You can see them right through the body cavity there. Those are our orientalis larvae. And so when a snake eats an infected prey item, those larvae will bore through the viscera, go straight for the lung, and then start the process of going through multiple molts before reaching reproductive maturity. And so once the male and the female, once mating or reproduction occurs in the lung, then the female will go through a final host to fully develop all of her reproductive organs, and then they will start to produce eggs, which are released in the lungs. And then we don't really know the mechanism of how they move into the mouth of a snake, but could be the cilia in the lung moving them forward, and then kind of, snakes don't have a diaphragm, so not necessarily a cough, but some type of action like that. And then it's swallowed and moved through the gastrointestinal tract. And then they proceed to go into the environment through the host species. And so how we detect them, obviously, if the snake is dead, it's really easy to detect them because they are very large parasites. They can be around 80 millimeters. So females on average that we see in our native snakes are around 50 millimeters. So they are quite large. You won't miss them. You can see an example on the left here. Fecal samples are the best way to detect infection in a live snake. So all of these circles here, this is under 10x microscope power. All of these circles here are RO eggs. Downside to the fecal samples is that we can't, and we don't know the morphological differences between pentasnove eggs. So then you have to go through the process of DNA extraction and conventional PCR. But I've done this many times and I've come up with optimal methods, so which are now published in our open access. So, and then colloidal swabs can be another way to detect it in live snakes, but they are about 20% effective. So we did colloidal swabs on a bunch of control animals, ones that we've dissected, and we found that it was about 20% effective. So not the best way to detect it, but snakes don't always give you a fecal material. And so I have a video in this, I can share the link to, so it can be linked to in the YouTube, but this is basically just showing, not this video on the right, but this video that has the link on my YouTube. It will show you that there is a clear difference in morphology between the native pentastomes that infect snakes, the porocephalus and the chirocephalus, compared to the Rescheduled orientalis. So the video here on the right is showing a black racer that had to be euthanized, and it has over 60 pentastomes, Rescheduled orientalis, in its lung here. And you can see after the host dies, they start to move out of the lung. So it's kind of, they're filling up a lot of the body cavity here. And so most of those animals are actually alive. But yeah, black racers definitely get some of the most severe infections. So now I'm going to kind of talk about what species here in Florida are at risk of infection and how far we think RO can spread. And so we knew that this was a problem in native snakes, so we're getting severe infections, and we knew that they could now, well, we had the knowledge now that they could infect a lot of different species throughout their life cycle. So cockroaches, small mammals, small lizards, small frogs, things that are often moved with humans, moved with plants by accident. And so we wanted to create a program to kind of monitor where this was going with opportunistic data. So we created, myself and Dr. Terry Farrell, we created SLAM, or Snake Lungworm Alliance and Monitoring. And I'm going to show you a little bit about the data that we've been collecting. And just a little bit about who we are. We have over 150 people involved now across the Southeast, and everyone is collecting either dead snakes that they find dead on the road or in habitat, or snakes that they are handling under their own permits and for their own research, and they're collecting feces or cloacal swabs for us. And the really cool thing about SLAM is that we're using opportunistic data, but we've been able to look at prevalence. We've been able to have new host records, new geographic expansion records, detections in the pet trade. And then using these samples, I was able to create a novel PCR assay that is specific to Relatella orientalis. So that makes it really easy to detect RO in feces and cloacal swabs and not amplify other DNA. We've also been able to publish a lot of our research in open access journals and engage with the community to try to just raise awareness that this is a threat to native snakes. And unfortunately, some of the snakes that we find are right in people's backyards. And so there just needs to be more research on this to figure out what the threats are, if it is a zoonotic concern or if it is a concern for our pet. And that's just showing here on the table the methods that we've created for the PCR assay are highly specific. So moving into who RO infects here in Florida, this was actually a black racer that I found right outside of my driveway, unfortunately, with a large female crawling on the pavement. But talking about intermediate hosts, a lot of this work has been done by Dr. Terry Farrell, whose paper will come out soon. But we have 8 different species documented here, with the 2 asterisks being invasive species. And so we have native and invasive intermediate hosts here in Florida, some of which are very widespread, like Anolis sacri and the southern toads. So for colubrids, we have quite a few across the state that we've found infected, with 2 new species being documented by SLAM reports. So the ribbon snake and then the scarlet king snake. And I have Drymarchon just bolded there because it is a federally protected species and we have found infections in them. For our water snakes and our mud snakes, or our dipsads here, we have found multiple mud snakes from South Florida with infections, which is in herp review. So if you guys need the PDF of that, just let me know. And so in total, we're going through all these lists, but there are at least 18 native snake species that are infected. And then we have our venomous snakes. Unfortunately, all the ones that you can find here in Florida are infected, including eastern diamondbacks, cottonmouths, pygmies, and coral snakes. And coral snakes had not been documented prior, but we have a paper that's in review right now with the Journal of Wildlife Diseases that highlights all the definitive hosts, all the new ones that we found, including Aspidocellus sex lineatus. So the race runner, we found adults, 1 in each of an individual's lung. So the definitive hosts aren't just snakes, unfortunately. And so talking a little bit more about that, there are at least 7 invasive species that serve as definitive hosts of RO, including the tokay gecko and tegus. And there are at least 5 invasive species that serve as intermediate hosts. So RO is a super generalist, which makes it even more of a concern how far it can spread. We've also found them in captivity. So I've found them in 5 different snakes brought into the pet trade, all of which, well, 4 of which died from infection. One was a water snake that was actually in Michigan that Dr. Terry Farrell found. And then we found adults in a bearded dragon, and this bearded dragon was held in an outdoor enclosure in Central Florida. And then an Aldabra tortoise who was about, I think, 3 or 4 months old and died of infection, and that was held in an outdoor enclosure in South Florida. So kind of get back to that later in the presentation, but there are a lot of concerns of animals in Florida in the Southeast being held in outdoor enclosures and either being exposed from wildlife or exposing wildlife to RO infection. And so just working off of the data from Snake Lungworm Alliance and monitoring, these are all the snakes that we have more than 10 or 10 or more individuals from. And you can see that the highest prevalence is really in the colubrid constrictor and the banded water snake. So black racers and the banded water snake. And kind of in comparison to that, if we check out some of these are, well, colubrid constrictors over 50%, and banded water snakes are getting close to that. And then if we take a look at the native pentostomes, only about 5% of the banded water snakes have been found with Chirocephalus coarctatus, and we've only found 1 individual in them. So if you remember, black racers can have over 100 pentostomes in their lung or RO in their lung. And we, with native pentostomes, we usually only find 1 with Chirocephalus and 1 to 11 with Porocephalus crotali. So the infection intensity with native pentostomes is a lot lower. And the prevalence is a lot lower too. I've only seen, these 2 infections of Chirocephalus coarctatus, pretty rare. Now, across regions, if you take a look at the map here on the right, this is what the map looked like for what we knew, where we knew RO was prior to SLAM, and then all of those dark counties here is what SLAM has found. So just through opportunistic sampling, we've just been able to identify where RO has been or is moving. And so we can see that South and Central Florida have really high prevalence, bumping around 40%, really, really high. And then in the Northeast, I believe it's about 16%. And then in the Northwest, it's about 5%. The only snakes that we've found infected in the Northwest has been 4 cottonmouths from 1 location. So that's kind of interesting. And when we look at our samples with the highest sample size, so the black racer on the left and the banded water snake on the right, the prevalence is quite high, and black racers are infected. We found them in Central Florida, South Florida, and Northeast Florida, so Northwest being just kind of that panhandle. I haven't gotten a lot of roadkill of black racers in the panhandle, so kind of biased there. They're probably infected if cottonmouths are. But the reason why I show this is that these might be 2 kind of indicator species of when RO has moved into an ecosystem. These are kind of what we're seeing as like the first animals we find infected. And for an example of this, one of my field study sites for pygmy rattlesnakes, I had found 2 or 3 black racers, 3 black racers that were infected with RO. And so, and I had at the time, and this happened over about 2 years, had not found any pygmies out of a sample size of over 50 that were infected. And just last month, we found a pygmy that was infected. So we do think, again, that these 2, colubrid constrictor, black racer, and banded water snakes could be kind of indicator species, given how high their prevalence is across the state. So again, just bringing it back, like, why is RO a concern? So we know that it has really intense infections in native snakes. It can cause pneumonia and sepsis, and it's associated with mortality and morbidity in at least 6 different species, and it might be contributing to population declines. And the species that are already in decline, like indigo snakes and southern hognoses, are at high risk of infection just based on their diets. So we see a lot of ectotherm eaters being the ones that have the highest prevalence, like the banded water snake and the black racer. And again, our naive hosts, our native snakes, have severe infections, up to 107 individuals within their lung. And here's just some examples of some we pulled out of water snakes as well as black racer. Another reason why we are concerned about RO on the conservation level is that it's likely just to continue its spread. So we know that their eggs are highly resilient. So if you look on the right here, these are just, this is just a graph of days after desiccation, and you can see that the dry eggs stayed about 80%, right? So all of those basically showing you that their survival. So basically what we did is we exposed eggs to different desiccation like time, so 5, 10, 15 days, and then we fed them to cockroaches, and then we saw that they still were able to infect those cockroaches. So desiccation does not kill the eggs, unfortunately. We also know that there's high prevalence and high severity and highly mobile snakes like black racers, and so they're moving to and from different habitats and are able to potentially be a super spreader. And then we also know that RO can infect a lot of intermediate hosts, both in diversity and ones that are super abundant. And they use synanthropic host species like brown anoles. So there's a picture of a brown anole here that made it all the way up to Boston, and cockroaches. So things that are easily moved with humans. Also, there's the threat of a lot of our animals here in Florida are held in outdoor enclosures. And so if they can either be exposed from wildlife or expose wildlife to infection. We also have evidence that's not published yet, of population declines, both in South and Central Florida native snake species. So I'll be sure to share those publications when they're out, but we are seeing associations with like the first detections of RO within a population and then subsequent declines there. And so I think I have just a few minutes to talk a little bit more about my dissertation. So I'm studying pygmy rattlesnakes, pictured here, so Cystrois malarius, across the state in Florida. And basically what I'm doing is I'm looking at the disease prevalence and severity, both with the invasive pentastome and fungal pathogens across 12 different locations, all the way south in the Everglades and all the way west in the Panhandle. And I'm looking at their immunogenetic diversity, as well as their whole genome diversity, to see if there are certain genotypes that are associated with susceptibility. So trying to get an idea if some animals or individuals or even populations can handle these threats better than others. And lastly, I'm looking at 3 locations specifically that I visit monthly as a mark recapture study to see how, and each of these locations has both fungal pathogens and the invasive pentastome. And I want to get an idea of how this is impacting survival as well as growth rate. Because unfortunately, we've found quite a few pygmy rattlesnakes dead in habitat with Rallitella orientalis. So getting an idea of survival in a small viper will be really important, and hopefully shine light on what the threat is for other snakes as well. So multi-pathogen surveillance, kind of wrapping up, like, what does this mean for us in the Southeast? I mean, multi-pathogen surveillance is definitely necessary in snakes, but we are seeing a lot of infections in captive reptiles, not just snakes. And so potentially this might mean that we need to restrict the trade of wild caught Florida reptiles because of how high that prevalence is in South and Central Florida, and how many just opportunistically we've found to be infected in the pet trade. So with that, we do have a website. The map needs to be updated, but our most recent publication that is in Biological Invasion Records has the most recent map. But if you have any questions or want to check out some pictures of more pentastomes or pygmies, you can visit these Instagram pages here. Yeah, and I guess I ended early, so I'm happy to take any questions.
That's a fascinating presentation, Jenna. Thank you very much.
Yeah,
Amazing how quickly that species has spread already since we first recognized it. I don't see any questions in the chat yet, but I do have one. So you mentioned a lot that, especially for like the black racer, the max you may see maybe over 100. But what's the mean or average of it, and kind of the range of results you might see, especially compared to the native pentastomes?
Yeah, so I mean, it depends on the species. So if we're talking about, like, for instance, corn snakes that are primarily mammal eaters, a lot of the time I see very low intensity where it's just like 1 to 5 pentastomes, but the range really can be from anywhere from 1 to 107 in our native snakes, and kind of the range and the mean differs per species. Trying to think, I feel like I have a table of the means, but yeah, so it differs between species, but I would say like the average that we've seen in black racers is probably between 30 and 40 individuals. And I think that's just based on how much black racers are actually eating, and so they just have a higher chance of consuming more infected prey items.
Do you think that maybe some individuals might have some sort of natural resistance that might be passed on to future generations and kind of help protect our snakes?
Yeah, I mean, I'm definitely hoping that is the case, and that's one of the reasons why I want to look at that in pygmy rattlesnakes to hopefully highlight that some snakes do have some type of tolerance or resistance level. Yeah, I mean, I don't know for sure, but I'm hopeful that they do, and that could be kind of leading into like maybe some populations that we've seen decline because of this parasite, they might be able to bounce back.
Well, yeah, hopefully we'll see that in the future.
Yeah.
You have any questions from the group? All right, well, I think that covers it. If you have questions, we've got her contact information that was shared earlier. And thank you very much, Jenna.
Yeah, thanks so much for having me. Feel free to contact me with questions.
All right, so I'll go switch gears now and talk a little bit about what we have upcoming here with our CISMAs and FISC, the Florida Invasive Species Council. So as we're hitting into summer, everyone's enjoying the heat, I'm sure. We're seeing a switch, not as many work days going on, but we still have a couple out there with some of the hardcore folks. But Treasure Coast CISMA and Central Florida CISMA are still getting a little bit of field work done as groups. Of course, everyone else we're all still out individually and doing a lot of that work. But we're seeing CISMA-wise a lot more kind of meetings coming up. So we've got several great virtual meetings statewide and with individual CISMAs. I do want to highlight next week, if you've got any sort of social media work yourself, or you just kind of want to get out, next week is Play Clean Go Awareness Week. If you're not familiar with that, they have some great materials that you can download to share out to kind of help people be aware of, just like Jenna was talking about, accidentally transporting invasive species around. So tips on how to clean yourself, your equipment, anything you're taking out in the woods with you, so that we are being less of an issue in spreading some of these invasive species ourselves. As far as announcements, the Florida Invasive Species Council's CISMA grant, we are still accepting proposals through the end of June 27th to be exact. You can find information on that at thefloridainvasives.org/grants, and I will also be including that information in the monthly CISMA Digest email that comes out later this week. And also want to make sure people are aware, we have a new working group coming up we're looking at developing guidelines for animal EDRR lists with the various CISMAs. And so that meeting is in a couple weeks, June 17th, the virtual meeting. You can email me for details, and that also will be going out on the monthly digest later this week. Looking at the upcoming calls, so later this month, we will have Jimmy Lange from Fairchild will be talking about the intersection of invasive plant management and rare species protection. And then we have a lot of other great topics coming on through the rest of the year. And that wraps it up for our call today. Thank you very much for joining us. Appreciate your time. All of these calls are available on our YouTube channel, and this one will be posted up there within a couple days. Thank you very much, and hope you all have a great week.